6.4. But the 6.1 and 6.4 sum up, but we'll see. So we talked a little bit about membranes. You're going to see that's probably the thing. Because if you think about it, that's what allows a eukaryotic cell to be, wow, that is a big glue stick. And I'm just gluing everywhere. Okay. That's what allows them to be compartmentalized, which is, I'm not going to write on that. That's got all kinds of stuff I drew. Let's start over here. But that's the deal. Like if you had to say, hey, what's your best feature, eukaryotic cell? It's I have all kinds of compartments. The only way you have those is by having membranes that allow those to be separated. So back to membranes, you have, if we go back and we think about it, what we drew, hey, maybe we can just do that. Here we go. So this is what you were drawing while we were talking about membranes in here. So the notes were, you have this double membrane around the nucleus, and it's continuous. Contiguous is another word. So it's attached to the whole membrane system within the cell. And initially, those membranes, they're all folded, which that's because of surface area, and that lets you get a lot more membrane in the same amount of space. But initially, they have ribosomes as part of the system, so that's why they're called the rough ER. And then the farther away that you move from, this seriously drives me crazy, farther that you move away from the nucleus, the fewer ribosomes you have, which ribosomes are going to be where proteins are made, and we'll talk about that, like how that works right here. But the farther you move, the fewer you have, until pretty soon you don't have any, and you have what's called the smooth ER, which that is just membrane. So if you think about what the differences would be, the difference, first of all, is location, farther away smooth, but also it's this ability to have proteins made right there where your membrane is being made. So what happens, we talked about this today. We drew, right? We drew this phospholipid bilayer, and we magically put proteins in the bilayer. Think about how hard that really would be. So you've got this big protein. How are you going to shove it into a membrane if it's traveling and you just plop it in there? It's not going to be able to be shoved in there because you've got to push. Like if I pushed this one in this direction, then the amphipathic properties, this hydrophilic part down here, when it was up here, I'd have to push it through the hydrophobic middle. How's that going to work? That's not going to work. So that's what the point is about having the ribosome right there where we're assembling these membranes. It seriously does make the protein and drop it into the membrane all in one step because it's sitting right there. So let's just write some, wow, that's my page real quick. Okay, let's just write some differences about smooth ER and rough ER. I think what I'll do, me and my favorite, just T-table. I like those because when you're studying, they're right there next to each other. So let's talk about the smooth ER and think about, like, what's the deal? Do they have different functions? Oh, yeah. We act like ER, whatever. Wow, curly hair, crazy thing. They're totally different. Just because you're made up of membrane does not make you the same. That's kind of like, walking over to, I don't know, a llama and saying you're made of cells, you're just like me. You're definitely not just like me. So just because you're made of membrane, they're not the same. So I think I'm going to start over here. and we'll just make some simple statements. Like, where do we find it? So it's going to be attached to, so continuous with the membranes around the nucleus. So with nuclear membranes. So it just naturally comes out of and flows out of this nuclear envelope. The number one thing that is the reason why it's called rough is it's going to, so it has ribosomes associated with it. So they're right there. That's very handy because it's going to also have, let's put these together. So this is going to be that it's going to make membranes Well, we had a big conversation. Why did we have to put proteins in a test question that didn't ask for them? It just talked about phospholipids. Well, it was because when we left our story, we had said the phospholipids, the way that they assemble themselves because of the water that's inside and outside of the cell, they assemble themselves in a way where that this hydrophobic middle pretty much means nothing comes in and out of the cell. Well, if nothing can come in and out of the cell, I bet it'd be real hard to get a protein in there too, unless you're assembling it at the same time. So the rough ER is going to have a ribosome right here that's going to make this protein, and it almost just drops it like, it sounds crazy, but like a rope through a hole in the floor. And it's going to come down in through here, and it's going to assemble itself inside this membrane space in a way that would make sense to be amphipathic. So you haven't got the membrane fully made around it. You drop it into this membrane that's being assembled, and it's going to line itself up along with how the phospholipids line up. You don't try to shove it in after the fact. So it's together. So this together means that you can have proteins put into the membrane with the phospholipids at the same time. And that actually works. That makes great sense. So you put the machine that makes the protein right with the place where the membranes are made, and then you just put them all together, kind of like making a sandwich. And they're going to go in according to their properties. So these proteins that are being made would have a primary structure that would have a whole huge run that's hydrophilic, a whole huge run that's hydrophobic, and a whole huge run that's hydrophilic, amphipathic. And then when you drop it in the membrane, everybody's going to be with everybody else that has the same kind of function, and it's going to be able, keep turning this page, be able to sit in the membrane in the proper position, because it's going to have this amphipathic part that's hydrophilic, hydrophobic, hydrophilic. And everybody's just going to get plopped in there all in a row, and they're going to coil up, turn in 3D, and get their tertiary structure because of hydrophobic interactions, and then it'll align with hydrophilic with the water. So that's one big job that the ER does. Here's where it is. Here's its job. Make some membranes. Well, membranes have proteins. We had to put that in our story because we have to explain that, well, no, they're not really very permeable if they're just phospholipids. Only little small nonpolar things go through. So we got to put some doors in there, put some proteins in there. So we said that it's going to make proteins that go in the membrane. It also, so let's put that in there. So makes membranes and makes proteins, one that go right into the membrane being made. So any membrane you have that has proteins inserted in it would be made right through here. But there's also proteins that don't go in the membrane, and therefore secretion. So it makes proteins that are also meant to be dumped out of the cell. That's what secretion means. So let's just put that. Just put dumped out of the cell. Now, one thing we don't talk about a whole lot, there's no magic in a cell. It's not like that when something is, let's say this protein's made and it's going to be secreted. It doesn't magically float where it's supposed to go and magically float out of the cell. Everything's pretty much trafficked, is what it's called. It's moved along either the cytoskeleton with microtubules, or somehow it is packaged and transported where it's supposed to go. So, we said it makes membranes. We said that the proteins that go in the membranes are made there by the ribosomes attached, and they get stuck right in there as the membrane's being made. Or, if you've got proteins that need to be dumped out of the cell for secretion, they're made and they're going to be packaged into a vesicle. The other thing that happens at the rough ER is you start adding tags to your proteins. So you're going to add a carb chain. They're short, 10, 12 monosaccharides. So add a carb chain to proteins. So at this point we want to call that protein modification. Yes. So we're just making that protein better able to do its job, because we're going to modify it by adding a carb chain. It's going to give it a tag. So, Smooth ER, what's your big deal? Well, I don't do any of that stuff. None. I know I'm the ER, but please. Kind of like you and your siblings. I know we have the last name the same, but please, we're not the same thing. So, it's going to be after the rough ER. So, we'll just put that. So, it's continuous. So, it's connected to the rough ER. Farther away from the nucleus. It has no ribosomes. Not its job. So it's not going to do, so let's think about that. Therefore, it is not about proteins. If you start talking proteins to it, it's going to be like, nope, go see my brother. Not my job. I don't even have the machinery for that. What it does do is it makes, so synthesizes, it makes lipids. Really? What kind of lipids? Well, my favorite. Going to make phospholipids, so those will go over here. That'll be part of making membranes. It also takes and makes steroids. So those are going to be things like cholesterol is a huge one. Well, that is also, if it's cholesterol shoved into the membrane, there could be different sex hormones. Cholesterol is the precursor for pretty much everything. So estradiol, progesterone, testosterone, cortisol, all of that comes from that, and they're all made right here. So that's a big part of what it does. Then, I think this is super cool. It's going to have a job where it converts toxic molecules into non-toxic molecules, but only in the liver. So if you're talking about skin cells and their smooth ER, that's not their job. So it detoxifies drugs, poisons, if the smooth ER is a liver cell. So liver cells only. I don't know, did you know that? Did you know that's what your liver's got like 400 jobs. That is no exaggeration. But a huge one is to detoxify the things that your body doesn't recognize as food. Well, unfortunately, that's a lot, because you're exposed to a ton of chemicals that nobody regulates. Hello, we don't know what those are. Goes through the liver. And you eat garbage you can't actually pronounce. Well, it's not food. It goes through the liver. Poor liver. No wonder we end up sick. It can't do its job. The last thing the Smoothie R does is it's going to store calcium ions. Well, who cares? Okay, let's go back to a word that I have told you I use all the time. It's the G word, gradient. If it stores calcium ions, that means there's more there than in the rest of the cell. So this becomes part of signaling. It's a way for us to use a gradient to do work. So this then becomes a gradient. Gradients exist. Something's higher somewhere, something's lower somewhere, so that it can do some type of work. Now we use them to actually make ATP, but in this particular case, the work is going to be information. Big deal. I would be able to do that, have that conversation. Okay, next part. That was 1. 2. I'm going over here. So we said there's no magic. So what about these proteins we make that we want to dump them out of the cell? Do we just go shoo shoo and they just know where they're going, they magically float out where they're supposed to? Nope. They are transported. So you've seen this picture a bunch where that, let's go over here. I don't want to draw it again. Let's go here. So you've got rough ER, membrane, blah, 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 smooth ER, blah, 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 Golgi. You get these flattened little disc-looking things, and they're the packaging place. So by the time you go through the rough ER and you're making protein, you're putting it in the membrane, and you're also making it for secretion, and then you move through here for the smooth ER, you kind of change the protein a little bit, you continue to modify it until it's just the way you want it, and then you push it off in its own little vesicle. So that's what this is. This is a transport vesicle. So if you think about the Golgi being a processing plant, think of it kind of like UPS. So the package, it's going to be produced, whatever you bought, blah, blah, blah. You get it from the factory. It gets boxed up here and packaged, and then it gets put on a truck, and the truck takes it where it's supposed to go. And we tagged it, so it's like putting a sticker on it, like a label. We tagged the protein, whatever it is we're going to secrete. And so the transport vesicle, based on the tag, will move that protein where it's supposed to go. It's kind of cool. I said there's no magic, but I don't know. That sounds pretty awesome. So this is how a transport vesicle is going to work. Let's walk through. First of all, I've got to work around that paper. Transport vesicle. So vesicle just means a small, like membrane, bubble, sphere, bag, compartment. Little small membrane, I don't know which one to call it, sphere. So it's wrapped in membrane, of course it is, because that's the only way this works. And you've tucked in there whatever you're wanting, this protein you've made for secretion. You've tucked it in there, and now the vesicle is going to literally move this to where that it's supposed to go, which in our case would be dumped out of the cell. So what's their job? Their job is to move, okay? Well, they have to also contain, so they're going to hold, package, and move different materials. What kind? Well, the proteins that we talked about for secretion, or that we're going to use for something else. Lipids we're going to use somewhere else. Oh, those right here, the steroid ones that we've made, so hormones. And the actual membrane that was made. So the vesicle is the actual membrane, and it just jumps in and joins the membrane where it's going. So you've got transport vesicles that are going to hold things, different materials, and actually going to become part of a membrane system themselves when they get there. So it's kind of cool. I mean, if you think about it, the packaging is part of what you're sending. So how's that going to work? So it's going to go from the ER, where we're making membrane, we're making proteins, we're over here, or we're making lipids and hormones, and we're going to take those things and we're going to go to the Golgi. Golgi is like the packaging facility. Make sure everything's modified in the way it needs to be. So where you're going to get final modifications. We're going to read the tags. We're going to see where they need to go. And then we're going to, because we said we were going to integrate these. Okay, so make final modifications for possible secretion. So it may be going outside of the cell or other cell locations. It's crazy that they get like an address sticker put on them with that whole carb tag. So then the way this works is they're part of the Golgi itself at first. So it's going to pinch off from the cis side. Now remember, cis means same, trans means across. So it's going to be, cis is the closest side to the nucleus, trans is the farthest side. So it's going to pinch off from the cis side, and it's going to move to the trans side of the Golgi. So there's a cis and a trans side of the Golgi. And you're going to have, through all this modification piece, you're going to modify the carb tags that are on the proteins. They're like the address label. And at this point, they're going to be called glyco. Glyco always deals with carb. Protein now, what we've made. And then if we need to, we'll do some modifications. So we're going to modify the phospholipids. And let's think about what that means. So you could literally change the head group out. You could change from phosphatidylcholine to phosphatidylethanolamine. None of that matters, but you could swap out, and I'm saying an enzyme would do this, swap out the head group. That's one thing you could do. You could swap out the different tails. You could change saturated to unsaturated, or back and forth. You could change short chains fatty acid to long chain fatty acids. You could do all kinds of different modifications of that phospholipid. Now that everybody's all the way they're supposed to be, at the final way they're supposed to be, then what you'll have is you'll have a vesicle, and that vesicle is made of the final phospholipids, like these right here. Yeah, I'm happy with that. I've made those the way I want them. So it's going to be made of the final phospholipids, and it's going to contain the final proteins. So I fixed them, I tagged them, I've got them the way I want them, I stuck them inside the phospholipid package that has the phospholipids that I made ready to go. And then all of that is going to move from, so the vesicle is all of this, is going to now leave the Golgi. It's like left the packaging plant. So it leaves the Golgi, and it's going to move to, and in this case we were just describing one that would have proteins that we were going to secrete and phospholipids that were going to become part of the plasma membrane. And so it's going to move to the cell membrane so it can dump those proteins out. So that's the way it's going to work. So we started up here. We've got different materials. We started in the ER by making the membranes, the proteins, the lipids, whatever between the smooth and the rough ER. Then you make sure that they were exactly the way you wanted them. Like you've made all of your mini modifications in the Golgi to make them exactly right for where they're going. So you don't want just any phospholipids. There's phospholipids in every membrane. Well, these membranes are going to the cell membrane. Oh, well, they need to be this type. So switch, fix by an enzyme. And then where are these proteins going? We're going to secrete them. Oh, they need to be like this. Tag, fix. And so then they get packaged inside that phospholipid sac of, here I made these, and they get sent to the membrane. So how does that even work? Like when they get there, how does that work? So here's what we are going to draw. Here's the Golgi. And we said in its final form, the vesicle pinches off, right? And then it's going to move to down here, which is the plasma membrane. Cell membrane, same thing. Okay, so what's it going to do? Like, it can't walk through that. How is it going to do its thing? And besides, the whole vesicle itself is actually made to become part of the plasma membrane too. Almost like an edible wrapper. So how do we do this? Well, this is the way it works. These are the steps. So I've got stuff in here, right, that I'm going to let go out of the cell. So this is the outside of the cell. So what will happen is that this vesicle will come sit on the plasma membrane, just like that. Then you're going to have an opening where what you secreted can then leave, and then this piece here is now just part of the plasma membrane. So since I have all these colors of pens, let's make this guy have a pink. You probably can't see that very good. So this is the new membrane right here, which means this is the new membrane. And then it's going to sit now in the old membrane, just like that. So the best way to do this, we make a piece of paper. And we're going to draw the two sides of the vesicle. So this is inside, okay? This is outside. So the vesicle looks like this, right? So let's see how that works. Let's see if that makes any sense. So if I have what would go on the outside of the cell membrane on the outside of the vesicle, and it travels down, the way this works is it kind of breaks open like an egg. Remember we drew this right here. And it would, come on, work with me, paper. It would pinch open and lay flat. Well, that's not right. That doesn't even make sense. Vesicles are inside out. What would you, let's think, first of all, we've got to talk about, like, sidedness. I know way I'm getting to that other strip. So, if you've got a bilayer, right, I'm not going to go crazy with it, I'm just going to do a few. And I'm going to put, you know, a protein that's all the way through, that's called integral. And I'm going to put some more phospholipids. And yeah, I know they're all saturated, but I don't know, okay, whatever. Here we go. Now I won't feel, I won't feel bad. All right, we got that? Just a piece of a membrane. There's sides. Do you agree? There's sidedness. This side, this is the cell membrane, and this is the inside of the cell, there's an inside face, so that's what this is. This is the inside face of the bilayer. And if this is the outside of the cell, this right here, this layer of the bilayer is the outside face of the bilayer. So, don't even act like this is complicated, because it's not. All you got to do is think about a house. For one wall, you have the inside of the wall that's inside the house, and then you have the outside of the wall that's on the outside of the house. Where do you put bricks? You got to put them on the outside. Where do you put your address label? Like, what is wrong with my hair? Okay. Where do you put the numbers that are like part of your house? Okay, I can't stand it. It's all in my eyeballs. Okay. Where do you put that? You need address number labels like on the inside of your house? Why would you do that? You know what your numbers are for your house. So would you hang your favorite picture on the outside of your house? No. You'd hang it on the inside. Where do you put curtains? You put them on the inside. So these are different. They're not the same. So you may have some proteins that are associated with one side of the membrane that are different from the kinds of things that are associated with the outside. It's called sidedness. So the kinds of things that are on the inside face are different than the kinds of things that are on the outside face. Like a house. There's definitely a difference with what you put on the inside wall and the outside wall, although the wall is one thing. It has a side that faces outside and a side that faces inside. So, let's see what we want. Wouldn't you want, here's my vesicle, the inside face of the vesicle to be, okay, I'm inside the cell, right? To be the part that's like interacting with all of the materials inside the cell. You want inside with inside. Well, will that work? You bet. Because when you're hiding the outside face, right? Because you might have things there that don't work with the inside. They're not supposed to be something that interacts with inside stuff. So you've got it tucked inside of the vesicle. So you're hiding the phospholipid outside face of the bilayer. And the only thing that's exposed to the inside of the cell is the inside face. So then as it floats down, and we'll let this right here, well, that right there would be fine, right? We're like right here, we're going to let this be the outside of the cell. This is going to open up, and when it merges, the inside face would be facing the inside of the cell, and the outside face is now part of, in the right place, with the outside of the cell. Because under here, all of this would be the outside of the cell. This is so much easier if I do it with you, which I will. But it makes perfect sense. So, the vesicles are inside out. So, for a vesicle, I'm going to so there's the inside of my vesicle, and here's the outside of the bilayer. So this is a vesicle. I'm going to color this part of the bilayer blue, this is going to be exposed to the inside of the cell, right? That's what's floating the way it is. And I've hidden, give me a good color, green. I've hidden the ends, this end right here, I've hidden this outside, going to eventually be on the outside of the cell. I've hidden it in here, along with the proteins I'm going to secrete. So as it travels to this plasma membrane up here, the inside part is going to, let's just use color now. Here's the outside of the cell. So this is what the plasma membrane looks like. So let's just make it this way. Okay, so here's our plasma membrane. I don't know if I can do two at a time. Ooh, look at me. I'm super proud of my drawing things two at a time-ness. Okay, so this is my cell membrane. So this is out of the cell. This would be, ooh, that's not a good arrow. This would be inside the cell. So far, so this is the inside bilayer. This is the outside bilayer. This is the cell membrane. So this is your phospholipid bilayer. And it has an inside and an outside. So if we really wanted to be weird, we'd just draw a few little phospholipids to remind ourselves what our colors were. When this guy comes here, as soon as this blue touches the blue, it's going to open up. So what you'll end up with is, you're going to end up with, here's your inside, here's your vesicle. Alright, that's what we have to keep see what I was doing. So the vesicle came and sat on the cell membrane. So the cell membrane originally was just this part. And when the vesicle sat, these that are the same, that'll be the inside face, are now continuous. And this part that you had hidden, when it opens up, it becomes continuous with the outside. And you're going to empty out the proteins. So then when you're done, it all lays flat. Ooh, I get to do this again. Whoop, I not do a good job. And there you go. So if the vesicle is from here to here, then this is the new phospholipid bilayer that is added to the cell membrane by the vesicle. The vesicle moved in and became part of the cell membrane. I think that's about the coolest thing ever. I think I'm going to stop there. How long is this? 39 minutes? They're supposed to be 45. Oh, I think I can do it. We'll do some more. It helps if you do your paper. But that's all about sidedness. There's an inside part of a piece, and there's an outside part of a piece. if you think about the membrane being a wall. It has an inside part and an outside part. It's a bilayer, and we have to make sure that we outfit each part for the role it's going to play. Inside's different than outside. Okay, way back here. Now it says, hey, there's a protein. It functions in the ER. It has to be modified in the Golgi before it can actually do its job. So we made it in the ER. Probably means it's part of a membrane. Then we're going to fix it, make sure it's perfect so it can do its job. And now we got to describe the path, starting with the RNA molecule that specifies the protein. Well, that's unfortunate because we haven't really done a whole lot of that. So, here's the way this works. The information, the directions for how to put the protein together are in the nucleus. So here's the DNA, and we're going to just have this DNA, and we're going to put that this is the gene that codes for the protein. And, well, you're not taking the DNA out of the nucleus. Whoop, nucleus. And so it's a double membrane. What you can do is you can make a copy of that information. I'm afraid you can't see the orange. Let's go to the ground. So you can make a copy of this gene, and that's messenger RNA. So this is your messenger RNA copy of the directions in the gene. So a copy of the gene. Well, DNA can't go out of the nucleus, but RNA goes right out. No problem. So there's pores in the nucleus, and they're too small for DNA to go out because it's double-stranded, but they're exactly perfect for RNA to go out. So this is your messenger RNA that you have the directions for how to make this protein that came from making a copy of the gene. Okay, that process right there is called transcription. So this is transcription. Now I've got the directions, I can make the protein. So you're going to have to modify that messenger RNA. I'm not going to do all of this, but we're just going to let it go on out. And I need to take it to the ribosome that's going to be on the rough ER, because that was in the directions. So this guy is going to go here, and that's the ribosome. The ribosome is the machinery that's going to actually allow us to read this code and to create a protein from it. So let's kind of blow that up. So let's come over here, show what that looks like. So this is just a blown up. This is enlarged. So what happens is you've got the small unit, subunit of the ribosome. It recognizes your messenger RNA, which is going to move this way. When these two together are there, they do what I said before, which is recruit. So it brings the large subunit. So this is large, this is small, this is the ribosome. Got the two parts. This is the messenger RNA. We're reading the code, and as we read the code, we're going to have the correct amino acids put in order. So these are the amino acids. And we're going to have the protein that was coded for by this gene. And we're here. This is where we are. This is the rough ER. We just blew it up. We're doing this right there. So while we're there and we're making membrane, we're going to plop this protein in the membrane as we make it. So you've got, so we've got this part going. Let's go to the next step. So you've got phospholipids that are being dropped in. We're making this membrane. So membrane is made. And then you're going to drop in the protein. Let's make it orange. You're going to drop in the protein into this, and it's going to then assemble itself the way it's supposed to be oriented based on the phospholipids going in around it, because it's also amphipathic. So now you've got a protein that was made when, I swear you didn't get to see any of that. So I made the phospholipids in there. We dropped in, so you're going to drop in the protein. It's going to come as it's made out of the ribosome. So this is the large and the small. The protein's going to come through here, plop in between as this is being made, and it's going to curl up in the way that the associations make sense. So just to keep our colors the same, it's going to align itself with polar goes with polar. That's not good. Non-polar goes with non-polar, which is hydrophobic. So that protein's amphipathic, and that's how you made it. So it said, describe the protein's path through the cell, starting with the messenger RNA that specifies the protein. Messenger RNA leaves the nucleus with the directions for the protein. It travels to the rough ER and goes and the information is read by the ribosome. Here we go. So ribosome assembles. It's going to have the messenger RNA move through it. Your protein's made, and as the protein is made, it's dropped into the membrane being made so that it can sit naturally in the membrane the way it's supposed to. So, there you go. That's what that answer is. Not in words, in a picture. So you would probably want to, let's do this to you do. You're going to want to put this in words. The path. That's a test question. From AP, it's been on 2 tests. So I'm going to put a big T there and a star. And I've lost my T. It's coming. That one's coming. Okay, I did it. Did I do it? Ooh, I'm right at 47. You're the best. Okay, I won't add anything on at the end. I think you're wonderful. Okay, you know me. I couldn't let you do this by yourself. So I'm going to show you a little clip, and then we're going to leave it up here where we can move it over. So maybe you can see it a little bit better. And then we'll decide how we'll put that in words. Okay. Let's see here. This one? Yes. So this is a little like piece of a video. It's really from like a med school study program. So a little bit beyond what we do. But it's really good at showing what's going on. So they are showing you that there's 3 steps in the process. So the first step is that you're preparing the membrane. So the membrane's being created, and there's a place within the membrane that's ready to receive the protein. So this is the ribosome. Here's the large subunit and the small subunit, and it's going to actually show it here. As it makes the protein, there's a tag on the protein that's like an address label. And that directs the ribosome as it's making the protein to the ER, and this spot, oh, sorry about that, and this spot right here that's ready for the protein to get dropped into it. So the ribosome makes the protein. Oh, hey, there's a tag. We're supposed to go to the ER, and we're going to insert as a transmembrane protein, which is what that's called, into this little spot that's ready for us. It's like parking your car, okay? So it's coming here, it's making the protein, it's being made as it's dropped into the pocket. And as it's dropped into the pocket, it's going to go in and it's going to fold within this pocket based on the properties that we talked about when we were doing the phospholipid short free response question together. So the middle part's going to be the hydrophobic part. These tail ends and loops are going to be the hydrophilic parts. And it's going to become able to just, of course, the ribosome will leave when it's done. And now it's going to be inserted and the membrane will be done. So it's all done together. And then we'll kind of draw this a little bit. Let me let him tell you what he's saying, and then we'll freeze it.
Here we'll learn how transmembrane proteins import into the ER and embed themselves in the...
See, I cut him off. I trimmed it so it wouldn't be forever. So let's just pause this. It'll quit doing this. And let's just draw what we see here. So we want to draw... Oh, it could help if I turned over here. Ooh, how am I going to do that? Hold on, let me figure this out. I guess that'll do. It'll have to do. Oh, technology. So we drew, look how that tore. I'm like a crazy person with pens. So we'll draw over here this left part, like I'm right here. So I'm going to draw that just right there. And I'm going to show that this is, and I'm going to add in, just because, you know, we need some reminders. There, so there's the phospholipid bilayer, which is what they're showing to the left. And I'm going to show this just kind of as a dotted line to make the point that, and we'll write it, that this is in the ER, so this would be the rough ER, because that's where membranes are made. Let's put here this membrane. Why am I writing this sideways? Anyway, this membrane is being created to be trafficked, weird word, which just means move. I don't know if there's a CK there, but I'm going to put it there. I like it. To be trafficked to the actual cell membrane. And it'll do that by becoming a vesicle. I could put becoming a vesicle. Alright, so this is going to roll up into a vesicle, get transported to the cell membrane, and do this whole thing, where that it just opens up and becomes part of it. So what happens is you've got the ribosome, and this is the protein that's being made, and it's going to be recruited based on the tag on the protein. So here's the ribosome with this protein, and this protein is going to drop in here, alpha helix. So it's being made. So the protein is made by the ribosome and dropped into the pocket. So this is just a parking space. What we call it that? Dropped into this parking space. That is made, how do I say that? It's made into the membrane. I'm using made a lot. Into the membrane being created. So we just left a spot there. And what you'll end up with is this membrane, like what we drew before, will be finished, and you'll have the protein. Now it's in the membrane. Because, you know, I talked about, and I didn't know how to really say it to you, so I can draw this a little bit like that, that you can't shove it in there once the membrane's made. It's the same reason it can't go across the membrane by itself. It's got hydrophilic, hydrophobic parts, and it just won't, like this part right down here, which was supposed to be a loop more like this, that's down here in the cytoplasm. It can't push through the hydrophobic part. So if I just shoved it in there, you have to have a place where it goes in while the membrane's being made. And so really what you're doing, if you think about it, instead of, hey, why can't I shove you in there because of all these awful forces fighting me? If you do it while you're building the membrane, those forces actually cause the protein to fold the way it should. You're using them. So you're going to exploit the natural, boy, my notes are all over the place trying to fit in this little square. I'm so sorry. Exploit the natural hydrophobic middle. Remember, if I told you to go hide, wouldn't you go to the middle rather than the outside? That helps you remember what's hydrophobic, what's hydrophilic. Hiding, phobia, you'd hide in the middle, not on the outside of something. And hydrophilic, outsides of the phospholipids in the membrane. So those are actual forces, and you're going to exploit those natural forces to cause the protein to fold. It's going to come together and fold correctly within. So I just put you can't see, WI / within the membrane. Okay, so I'm going to hush and let this just sit here for a second so you can catch up.